Hello, everybody. I'm Catalin. I'm going to speak a bit about functional paths, recording functional neural data towards WBE. Don't really know what emulators are, but I'm open to exploring this with you guys this weekend. This week, sorry. I'm open to talking about a bunch of things, including field development, nonprofit, essentially community. I'm a systems and behavior neuroscientist, so anyone from CS maths, please reach out if you have interesting ideas. Definitely interesting foundational models for WBE and what the heck emulators are. Today I'm going to talk a little bit about motivating in vivo neuroethology, the approach that I'm going to do, the data set size is speaking of motivating industry, a bit of a theoretical framework of emulator theory that I just published and advertised that I'm going to be at Lab Week next month. Thank you for the sponsorship of Foresight. Yeah, quickly. So this approach is pretty simple. It's pretty obvious to everyone probably in the room. I don't really need to give you too much of an overview. Basically, I'm seeking to record rodent electrophysiology, neural states, and behavior, build a neural emulator using probably something like a language model, and then build behavior predictions and neural state predictions. Okay, this is pretty obvious to everyone. And what might not be obvious, and this is the thing I'm trying to work on, is if modeling neural dynamics is sufficient to create whole brain emulation, skip entire mechanistic neuroscience and connectome. And this is this paper that I submitted to NeurIPS. I'm going to share with you probably after. I take a data types viewpoint at WBE. We can look at WBE from a bunch of different perspectives. You know, we really want to capture dynamics, but maybe not. Seeing Nathan's talk, I'm not quite sure if we need to go all the way to like the nanosecond scale in terms of speed and size. There's a bunch of trade-offs. And all these recording modalities like extracellular EFIS, which is what I work on. You can get 10 kilohertz, 30 kilohertz, et cetera. Capturing complexity, this is something people don't think about maybe as much. Maybe we should think a little bit more. And what I'm very interested in knowing is where's this asymptoting, you know, as we measure in vivo organisms, biological organisms, will the complexity asymptote, right? So we have an animal in the lab already doing some tasks. Probably you have most of the complexity or not. Is it go this way? Do really the long tail distributions of these natural dynamics? It's a very, very complicated question to ask. To answer, capturing the whole brain, this is obvious. You know, electron microscopy is amazing, but you know, we need to really scale it up. Yeah, maybe we need some fancy terms like physiomics and holistomics or something like that for WBE. So 3 framing concepts for my research are, one of them is spatiotemporal scale. I like to do in vivo neural recordings at some scale. My approach is sparse, somewhere around local field potential, somewhere maybe some single units. But the idea is to fit in some empirical data points. Speaking of selling this to industry, what the heck do we need to build things that are increasingly impressive and predictive capabilities? That's why I like to put those points on the map. Happy to talk about, by the way, Facebook and TikTok are somewhere here. They're just looking at social behavior and they're doing a pretty good job monetizing that. One thing that people don't really think about, I think, as much is that neural dynamics are extremely complicated. And when you look at, if you look at a neural network, put in all the possible states, you shuffle the data, it's a huge, huge space. The real brains don't really spend any time in this in giant space. They spend time, the brain, spends time in very, very small space. When you do stimulus evoked, it's a very small space, and then spontaneous activity is much, much bigger. And I'm very, very interested in spontaneous neural dynamics. And this is my area of expertise, which is neuroscience of agency. We've known for 30, 40, 50 years, something weird happens when people make voluntary reactions. The brain kind of builds up a readiness potential or something like that that's quite subconscious. It's really, really fascinating, but I really think we need to capture this to understand whole brain emulation as a field. This is obvious to everyone. We might want to replicate the success of language model. Can we create enough data to just throw data at language models that will help us? All right, so that was a little fast, but my approach is pretty straightforward. It may not be this complex. I'm thinking about putting rodents, the plans to put rodents in some kind of open field environments and do electrophysiology for thousands of hours. With environments can be increasingly complex with tasks about self-agency and self-causality. People work on ethology, please reach out, or consciousness and agency, please reach out. I'm happy to talk. Existing tech is pretty much there. 10 years ago, I wouldn't have even thought about this. This little chip here can cost $1 million to make, but there's a few of them already. Fantastic, super happy. I just have to innovate a little bit, and I can already get to work. My 1-year milestones for this practical work I'm planning on doing is to just record from maybe 50 to 100 brain areas for something like 30 days and generate nice data. And the longer term scale, of course, is scale up, and see if people are interested in supporting this type of work. What really is the goal is towards the 1 trillion neural data points. Mark, I'm sorry for the amateurish spreadsheet, but basically one mouse, if you record for 1 month, you get to around 2.5 billion time points. This is a 1 millisecond time. It's about 2.5 billion milliseconds in a month of recording. If you actually multiply by the number of brain areas, for some reason, if you want to conceptually think, it's around 100 billion. In about a year and a half, you could reasonably record from about 30 mice. You're getting to the 2 trillion data point mark. Rats is very similar. 10 rats, they can actually handle much more larger neural arrays. You can probably do 500 now-ish. So you're talking about the trillion point range and maybe 100 billion time point range. Again, these are interesting things to think about and talk. I'm really fascinated also about the neural lifetime. Can we actually record for an entire lifetime from a runner for a couple of years? Why am I talking about this? Well, it's because I like to think about what these data set sizes look like compared to what it took to train a very successful language model. We didn't fit any kind of mechanistic, mostly no mechanistic heuristics into these models, and they seem to be pretty fascinating, pretty amazing results, and they're around 10 to 12, 10 to 13 data points, and that's what those data points look like. Is this data enough? I don't know. We need points on this distribution. Are there still conceptual elimination? Most likely. I think most neuroscience will say you need some mechanism. You need to have a whole bunch of heuristics. You just can't explore the space with data, but we need to build this data set to understand those. The theoretical framework, I was interested in 2 questions, how to build emulators, and I spoke briefly about this paper, the practical approaches to doing that, and in this paper. And one thing I do want to mention today is whether emulators can be conscious, which I'm really fascinated about. In a previous life, I was going to become a philosopher of physics and interested. I was very fascinated by causality. And this is a very interesting statement from Sandberg and Bostrom, the original paper. If you can mimic the internal causality dynamics, you have something that looks like the original organism. And in my paper, I talk essentially about this. Like if we can if we can model the neural dynamics of a system, biological system, we observe it, we understand it, we model it, and then we drive either the same system or some other system using a supercomputer or something like that. Causality, the input-output structure, and the causal. I'm done. Okay, and the output structure. Okay, sorry. And the output structure of the model is captured by the supercomputer, and it's very difficult to tell the difference indistinguishably empirically from the two. From the biological organism and the similar organism. And this is what this essentially conjecture says, that maybe you don't need mechanisms or the connectome to emulate brains. This is a very quick side note. I think these data sets will help the connectomes people. I'm happy to talk about it, how to carry it. I did these supercomputer simulations when I was at the Allen Institute. You get data that looks like real biological data. And that's it. Thank you.
Awesome. Thank you.
So I was wondering, I guess you mentioned a couple of times that this kind of data could surpass or circumvent the need for connectomes. Maybe I missed something, but I'm not quite sure I see how that happens. So could you elaborate a little bit on how that occurs?
Yeah, so it's obviously much longer. So it's a question, what is an emulator? What is the goal of the emulator? If you want to make a copy of the individual, if you can model and emulate their behaviors and have a system that kind of generates those behaviors from something like it's like the neural dynamics of that system you've organized. So there's a constraint. We're not just throwing language models unconstrained. The language models model the input output and the neural dynamics, right? So something like that. But it does depend on what your definition of an emulator is, right? And this is what I think hopefully some of us getting together, we can try to figure out what that is. Yeah, I'm happy to also talk, of course, after. Yeah. Sorry, who's next? Yeah. Yeah, no, please go ahead.
So there's a fallacy in the big data.
Yes.
You're showing all that increase of data that is happening with thousands of electrodes that you're recording. But what you're doing is with thousands of electrodes, you're sparsely sampling a system that usually encodes information sparsely.
Yes.
So you have lots of neurons that are doing nothing in the brain, that just fire willy-nilly in the brain all the time, and you'll be recording of those.
Yes, I hope so.
The neurons that do the important information, you will not be recording from.
Yes.
When you're sampling this thing. So is there a different approach? Is there an approach where you can actually record important information?
First of all, I'm an evolutionary biologist at heart. I don't think there's such thing as a non-important neuron in the brain, but I think more fundamentally is, I didn't have time to talk about this, but this is a scalable approach, right? And I'm not recording thousands. I wish I could record thousands. We're recording probably hundreds of sites. And the idea is that we accelerate, we show what can be done, and the next generation was like, okay, we need to accelerate neural dust. We need to put these little micro receivers in the brain, inject them in the blood. It's just like, it can be done. I think, I don't want to poo-poo academia, but we should be doing this from this side. We really need to stimulate the imagination of academics, I think. But you're absolutely right. There's a lot of redundancy and a lot of things that we don't understand about the brain. Yeah.
We have to close it here.
Okay, thank
so
you.
much.